Good morning, all. My voice audible or not? Voice clear? Am I audible? Hello?
Yes, sir.
Right. Today, we are going to discuss about another important topic about gymnosperm characters. So, what are gymnosperms? Already we discussed in practical training classes also. What are gymnosperms?
Plants which are having naked seeds.
Naked seeds, right? So, they are called gymnosperms. So, before that, we learn about the bryophytic character and pteridophytic character and type studies also. So now we are going to discuss about gymnosperms. In this gymnosperms, important type studies, Pinus and Natum. So including general characters, we learned about Pinus characters and Natum character. So anyway, here if we, for example, directly enter into the Pinus type study, are you able to see this slide? Hello? Are you able to see this Pinus distribution occurrence? Hello? Is it visible or not? Hello?
Yes, sir. Visible.
Right. First of all, uh, in general character or any type study, just wait, by this general character or any type study, first we discuss about distribution and occurrence. Distribution occurrence. Let's see here. Right. Mainly the gymnosperms. Of course, we are now discussing about pinus. Not only pinus, all type of gymnosperm mainly distributed in northern hemisphere. Northern hemisphere mostly temperate areas. That is about common gymnosperms. Northern hemisphere region, that mean, for example, this is the globe, equatorial line. This is northern hemisphere. This is southern hemisphere. So mainly the gymnosperms located at northern side, right? Majority of the northern side. And in India, they are mostly found in the region of Himalaya region. So that is the distribution of these gymnosperms. Like that, here the pinus and some netums also same distributed the region of northern hemisphere, mostly temperate areas. In pinus, there are 90 species. 90 species are found and six Indian species are recognized and worldwide 90 almost. But India, Pinus excelsa, Pinus long. This is important for short answers. Write about the Pinus Indian species. So just you write the six species: Excelsa, Longifolia, Gerardiana, Insularis, Armadi. So these are the Indian regions. As I said, they are mainly found in Himalaya region. You must have seen and some films also these type of pictures. Most of the films, these we can see these conifer shape plants, especially the pinus trees. Let's see here. So these are the pinus tree. Like this is the pinus tree longifolia. This is also Pinus roxburghi. This is insularis. So these are the different type of pinus trees. You must have seen during practical training classes also pinus male cone, pinus female cone. Right? A systematic position, gymnospermae, coniferaptida, coniferapsida, coniferal spinaceae, pinus. This is the classification. Now let's see the morphology. Here already you know the main plant body sporophyte. But in primitive plants, main plant body gametophyte. What you see in your naked eye, that is gametophyte. But here advanced means maybe some pteridophytic character. Pteridophyta main plant body is sporophyte. Like that, gymnosperms also main plant body. Gymnosperms also main plant body is here sporophytic plant body. What is our external appearance? That is called sporophytic plant body. So the simple language in our natural general knowledge and language, that is called a tree. So let's see what are the parts of the tree. The tree should consist of root system and stem and leaves. Let's discuss one by one. The roots well developed and taproot system. The same in Pteridophyta root system present, but it is not a well developed. Instead of taproot system, adventitious roots in Pteridophyta. If you take any Pteridophyta, and stem, main trunk is unbranched. Main trunk is unbranched. It has branches confined to apical region. Branches are two types of branches, that is called dimorphic. These two type of branches called, one is unlimited and long shoot. Branches limited and short shoot dwarf are called as spurs. The leaves, foliage leaves means green leaves are present. Another one is scale leaves are present. For example, if you take the rough diagram of the plants, this is the main trunk, stem, main trunk. No, it is unbranched. It is unbranched. Actually, main trunk. This shows continuation growth. On the main trunk, two type of branches here. It is instead of unbranched, actually not much branched. Only two type of branches on the main trunk. The two type of branches, one is unlimited or long shoot. So this is long shoot. So that means it shows continuation growth. On the long shoots and limited means, so this is extended from till formation of the foliage leaves. Like these are the foliage leaves. After formation foliage leaves, the growth is retarded. So that's why this is limited and this is unlimited. On the if it take the for the leaves, two types of leaf, foliage leaf and the scaly leaves. For example, this is the foliage leaves. Foliage leaves are in green in color. Let's change color. Foliage leaves green in color, and they are only found in la, dwarf shoot, or limited shoot, and also some scale. Let's change color. Apart from that, scales also present. Sorry, scales also present. Apart from that, scales also present. In long shoot, only scale leaves are present. Yellow color, this scale leaves are present on long shoot. Like so, only two types of branches, two types of leaves are there. Dimorphic foliage, or another name is needle scale leaf. Right. This is about the body structure. This also you know already. Next one. So main plant body also, sporophytic plant body also clear. Right. I will explain about this cone, male cone, female cone. Already you know about in practical training classes. Again, I will explain later male cone and female cone. Internal structure. Internal structure also, this is only the important one is TS of leaf, needle TS. Yesterday itself, you demonstrate the section cutting of pinus needle. Most of the students done very well about this section cutting and clear triangular outlines with vascular bundles. Already we discussed yesterday during practical training classes about the needle internal structure. I don't waste your time and my time. And another important, but here remember this is TSFly very very important for short answer. Next, reproduction in pinus. This is very important. Reproduction in pinus. What is the reproductive part? Reproductive part in gymnosperms. In gymnosperm, reproductive part is cone. In pteridophyte, a reproductive part is cone. Now, here also reproductive part is cone, and it is the first point here given clearly monoecious. What is monoecious? What is dioecious? Hello? What is monoecious? Kusuma, Vaishnavi. What is monoecious? Kusuma, Vaishnavi. My voice is audible or not? Hello?
Yes, sir.
Right.
Yes, sir,
Not
but I don't know, sir.
the clearly given in the slide visible or not? Reproduction in pinus.
Yes, sir. Visible.
Right. So that means the sporophyte plant body is monoecious. But the male and female cones are produced on separated branches on the same plant. Mono is a dioecious means male and female separate. That is dioecious. Monoecious means the male and female developed on same plant. If the male and female developed on same plant, that is called monoecious. Here it is monoecious, same plant but different branches. Different branches. One is a dwarf branch, one is long branch. Let's see first of all that mean male cone separate and female cone separate. First we discuss about male cone, later we discuss about female cone. The male cone, it is mainly the originated the lower branches of the axial scale of leaf. Axial scale of leaves, lower branches appear in the month of the January in plains, but hill region mainly in March month reaching maturity. They can be seen clustered just behind the shoot apex, like this, like a cluster. This is group of male cone, group of male cones raised. And another one is this is the axil of the scales and the place of the dwarf shoot. This is dwarf shoot. This is dwarf shoot. This already again dwarf shoot. The place of the dwarf shoot replaced by the group of male cones. Same the female cone upper branches of tree axil of the scale leaves either single or group. Here the male cone small as a group. Almost it is group, male cone group. That is compound cones. But what about here? A female cone. The female cone, the bone upper branches of the tree, axils of the scale leaves, either single or groups. Maybe, of course, it is even group just two to four. That's it. But male cone more than four group. But female cone only two or four. That's it. Male cones seen in the month of February get pollinated in 3 to 4 months. The complete maturation and seed dispersal takes place in the third year, in the third year of the plants. And when an actual not given year, and remember, when compared with male cone, female cone large in size. That is another important. Origin also different. Origin, the female cone developed on long shoot. Male cone developed on uh, dwarf shoot. Female long, male dwarf. And the female one only 2 to 4 as a pair, is a pair. But male not a pair, is a group, aggregated group. And when compared with size, male cone small, female cone large in size. So that is the difference between male and female cones. Next, microsporogenesis and male gametophyte development. This is important for long answer. Microsporogenesis and male gametophyte. And before that, actually, one word forget here. How the cone made up of? All that told you. How any cone, whether it is male or female cone. First, you tell me the cone made up of. Hello, hello.
Yes, sir.
Any cone made up of. Already the same discussed yesterday. Classes, practical classes also. This is cone LS. Now this cone, this each one considered as this each one, this one, this one, this each one considered as. Anyway, remember the first of all cone, the group of sporophylls altogether form as a cone. What is present inside the sporophyll? Leaf and
Spores,
sporangium.
leaf and sporangiums.
Right, sporangium. What are present inside the sporangium?
Spores.
That's it.
Spore of spores, yes, sir.
Spores. So this is commonly happened. But in pinus, male cone, not a simple cone. In pinus, male cone, female cone separate. The cone sporophyll. What about male cone? Male cone made up of which leaf?
Microspores.
Right, microsporophylls. Like here,
Spore.
microsporophyle. What are how it is made up of microsporophyll? What are present in microsporophyll? Sporophyll.
Microspores.
Microspores developed inside sporophyll.
Spores.
Here the spores developed inside spores, sporangia. Microspores developed inside the microsporangia.
Yes, sir.
So next microsporangia. Microsporangia like this two microsporangia. So inside microsporangia, microspores are done. Now let's discuss microsporogenesis and male gametophyte. The simple, the spore genesis, production of spores called as sporogenesis. Here the production of microspores. Production of microspores called as microsporogenesis. What happened after formation of the spores here generally? The spores further developed into
Gametes.
right
Gametes.
gametophyte, gametophyte,
Gametophyte.
gametophyte. Spores gametophyte. What about the microspores? Here microspores, microsporangia, microspores which produce microsporangia. Microsporophyll consisting microsporangia. Microsporangia it produce microspores. So that is called microsporogenesis. Production of microspore from microsporangia called microsporogenesis. Then after formation, this microspores further developed into it the spore further developed into gametophyte. What about microspore? Hello? Heading itself given. That mean development as a spore.
Gamete.
Which gamete?
Male gamete.
Right, male gametophyte. First male gametophyte like this. Male gametophyte. From male gametophyte, male gametes are developed. Let's see how the male gamete developed. So before that, the development of microsporangia, the microsporangia development use sporangiate. So here the two type of development that is leptosporangiate and use sporangiate. The use sporangiate here, let's see the use sporangiate development within the microsporangium. The microspore mother cell undergo meiotic division and form haploid microspores. Already you know that. Uh, Eusporangiate only single cell development of microspores from the single cell that is called eusporangiate. Group from the group of cells that is called leptosporangiate. That is the difference between eusporangiate and leptosporangiate. Right. If let's see here microsporangia, microspore mother cell MMC, microspore mother cell meiosis, microspores. After microspores, male gamete developed. Simple. Let's see diagrammatically. Already here the cone. This is cone LS. So each one considered as a microsporophyll. Each microsporophyll having microsporangia. Inside microsporangia, microspore mother cells. For example, this is microsporangia structure inside microspore mother cell. This microspore mother cell undergoes to meiotic division. As a result, formation of microspores like this. These are called microspores. After formation microspore. This microspores structure single cell winged like this, winged. So this is microspore. But continuation diagram, this is the continuation. What happened? One division. Inside this is first was one nucleus. This one nucleus divide mitotically and form prothalial cell and central cell. Two celled structure like this. Let's draw here. Two cell structure and rest of the common wings. Two cell structure. Again divided. Next again divided and second prothalial, first prothalial, antheridial initial. Three celled stage. Later again it is divided and it is again divided into first. That means again after central cell divide mitotically, form second prothalial cell antheridial initial. Antheridial initial. Released from the sporangium at set ovule through the wind well at the three-celled stage. What are the this stage? This three-celled stage or sometimes four-celled stage. Now this is gametophyte. The already after that let's draw here. This next diagram. This diagram, clear diagram. That means this central cell divided, two cellulars formed. Already here the first prothalial, after division of this cell, second prothalial, antheridial cells are formed. From the antheridial cells, male gametes are developed. From antheridial cell, male gametes are developed. So this is the development of male gametophyte. Male gamete, right? Next, next like that, microsporogenesis. Now the megasporogenesis and female gametophyte development. Let's see megasporogenesis. Now, where the megaspores are formed? Hello, hello.
Yes, sir.
Megaspores are developed inside microspores.
Megasporophyll.
Right, megasporangium. Right. So the group of megasporophylls combined form as a simple.
Megaspore.
No, no, no. Which cone? Female cone. Female cone. This female cone made up of megasporophyll. Actually, this megasporophyll is the compound. Compound means it is this sporophylls two types: bract scale, ovaliferous scale. Ovaliferous. Bract scale, ovaliferous scale. Ovaliferous scale, in name itself given. Ovule or megasporangia. Megasporangia or ovule. Now, what are developed inside the megasporangia or ovule? Hello? Hello? What are developed megasporangia or ovule?
Megaspores, sir.
Right, megaspore. So that production of megaspores called as megasporogenesis. Right? After the megaspore formation, this megaspore further developed into megaspore, further developed into, already told you, spore further developed into microspore, further developed into microspore, first of all.
Gametes.
Which gametes?
Male gametes.
Female gametes.
I'm asking out microspore. Microspore
Male.
male.
Male gametes.
Megaspore
Female gametes.
female gamete. That before formation female gamete, gametophyte developed as a female gametophyte. Let's see female gametophyte development and megasporogenesis. Already you know megasporogenesis, production of megaspore, how they are formed. So the cone in maturity and usual cylindrical, it is not a important characters. It has already you know that already you must have learnt in during practical training classes. It has central axis bearing spiral arranged oviviparous scales. Already told you oviviparous scales with the bracts scales. Oviviparous scales. Each oviviparous scale has two ovule. This is important. How many? Two ovule at upper surface of the leaf. Right. So this is female cone. LS of female cone. Two ovules. This ovule clear structure. Megaspore. Pinus ovule clear structure. Let's see inside ovule what happened. Inside ovule, nucellar cell differentiated. That means if you take in ovule, first of all, this ovule, two types, integument, nucellar cells. This is integument. This outer, this complete integument, this complete portion. This is integument. This is inside nucellar cells. Nucellar cells. Now this integument, three regions: outer layer, middle layer, inner layer. Outer layer, middle layer, inner layer. And this opening of this ovule called as micropyle. And rest of the place, these cells are called nucellar cells. From nucellar cell, what happened? This nucellar differentiated into megaspore mother cell, MMC, megaspore mother cell, like a microspore mother cell. Here also my megaspore mother cell. This megaspore mother cell undergoes to meiotic division. As a result of four megaspores are formed. So three megaspore degenerate, degenerate, only one active. Now this is the functional. So now this functional megaspore further developed into female gametophyte. Further develop female gametophyte or endosperm. Like this female gametophyte, this is female gametophyte or archegonium developed. Female gametophyte archegonium develop inside the ovum or megasporangium. Like this, how many? Two archegonium, two megaspores, two endospores are developed inside the ovum. Inside the archegonium, female gamete developed. This is this one, female gamete developed. Now fertilization. What is fertilization?
Fusion of male and
Fusion
female.
of male and
Right.
female gametes.
Already we know the development of male gamete, how it is developed, and development of female gamete also. Now already we have seen. Now fertilization. Now the fertilization here. Pollination, fertilization. What is pollination? What is fertilization?
Pollen grains reach to stigma to form a tube.
Right.
pollen grain.
Formation, that mean the pollen grain transfer one place to the another place, such as stigma, that is pollination. After pollination, male gamete fuse with female gamete, that is called fertilization.
Yes, sir.
Right. So pollination, wind pollination, that means pollination takes place by wind. Then fertilization, what happen?
Zygote is formed.
Zygote
Right,
formation.
right. Here you can see the zygote. This zygote developed into embryo.
Yeah.
Embryo less further developed into seed and developed into sporophyte. So that is alternation generation. Alternation generation, this life cycle important. So alternation generation means, first, for example, this is sporophytic plant. Let's take pen. What is sporophytic plant body? We start the class from sporophytic plant body parts: root system, stem, leaf, reproductive part is male cone, male reproductive male cone, female one female cone. For example, this is male cone. Male cone made up of microsporophyll. Microsporophyll have the microsporangia, which produce the microspores here. Microspores. Microspore further developed into this microspore, further developed into male gametophyte. This side. Male gametophyte, actually not given. Male gametophyte, which produce male gametes. Male gametes. Likewise, here also female cone, oviparous scales made up of megaspore mother cell, megaspore, which is further developed into female gametophyte or archegonia, like this. After fertilization, zygote, embryo development, seed, and again seed germination to sporophyte. So this germination called as sporophytic alternation generation. In this one is sporophytic stage, this stage sporophytic, in this is gametophytic. Spore, after spore production of gametes, that until the zygote formation, that is sporophyte gametophytic stage. From zygote to development of the cones, that is sporophytic stage. That's it. Next one. Draw this important. Take a picture. Life cycle important.
Yeah, done, sir.
In examination, if they ask about, write about gymnast general character also same you can write. Same right, but no, did not mention any penis, any needle. Simply, if they ask about gymnosperm character, you merely write about gymnosperm. Gymnosperm, sporophytic plant body, root cyst. Gymnosperm's reproductive part is male, male cone, female cone. Development as a male gametes and female gametes. That's it. That is about the general character. Now let's see. uh, So economic importance, no need to explain. It is, you can read yourself. Economic importance of the pinus tree and economic importance of gymnosperms also important. Gymnosperms economic important also very important. You can see in the book. There is no scope to explanation because just read the headings. What are the importance of gymnosperms? Like this here also some important given. Right. Next one, neatum. Another important one, neatum. Let's take PPT. Neatum, actually this is advanced gymnosperms. Neatum is advanced gymnosperm. Almost it resembles to the uh, angiospermic plant. Neatum, is it clear? Visible or not? PPT?
Yes.
Right. So like Pinus systematic position, same class, Neetopsida, Neetales, Neetaceae, Neetum. First heading in Pinus also distribution heading. Here also distribution heading. It is consists 30 species widely distributed In the world, tropical, already told you, tropical and subtropical zones, most of the species are lions. That what is lions? Lions are woody climbers that are called lions. Some common Indian species. Here, Indian species important. Indian species of pinus important. Indian species of neem important. Here are some Indian species: Netum mula, Netum neeman, Netum latifolia, Netum montanum, contractum. They are the some Indian species. The Netum represented about the 40 species confined to tropical and humid region. Nearly all the species except microcarpum occurs below the altitude 1500, and five species Netum contractum, Neeman montatum have been reported in India. Already we discussed. According to Bharadwaj scientist, various species of Netum occur in India following regions Netum bula. Woody climber, already told you lions, that is called woody climbers, right? Netum bula, it is woody climber having branches with the swollen. Actually, no need to explanation about the different plants. Just remember the names: Netum bula, contractum, Netum neeman, Netum latifolia, like this. Now let's see sporophytic plant characterization. The most of the species are climbers, except being shrubs and trees. Two types of branches like a pinus. Here also same limited branch and unlimited branch. The climbing species have the branches of limited growth. That means short growth. Leaves dicot like large oval margin. If you take pinus, they are not well developed leaves. They are needle like structure. But if you see the netum clearly, these are angiospermic leaf, angiospermic leaf, like this dicot angiospermic leaf here, like this we can see. These are the leaf, and not given here. The leaf arrangement also opposite decosit, opposite. These two opposite and these two opposite. So, but different direction. That's why this is called opposite decosit. And it has the reticulate venation. Reticulate venation is the characteristic feature of dicot leaf. Anatomical structure not important one. Stem anatomy important here. Stem anatomy important for short, long answer. Stem anatomy important, and abnormal second growth of the needle also important. Abnormal second growth and anomalous second growth important. If you take any internal structure, whether it is important or not, but remember, any internal, very internal, three parts are there. That is called outline, epidermis, and cortex and steel. So here the single always epidermis single layered. You can see here outgrowth, sunken stomata present. They are the character of epidermis. And cortex also, it is actually not a single layered. It is more than say, one layer, 5 to 7 cells. Is made up of chlorenchymatous region, a middle parenchymatous region, and sclerenchymatous, inner sclerenchymatous. So different type of group of cells are there in cortex. Next in steel, mainly in steel, xylem present and phloem present. The xylem consists of tracheids and vessels. Present vessel and angiospermic character. Actually, the vessels absent in gymnosperms and pteridophyta, but some advanced such as natum, welwitschia, in xylem, vessel cells are present. That much enough. Next one. And actually not given here. And arrangement of the vascular bundle, U-steel type. U-steel type also one of the important character, advanced character. U-steel means the appearance of the vascular bundles form as a ring. This vascular bundle collateral, conjoint, and endark type of vascular bundle. So this is about internal structure of the stem. Now reproduction. What are the reproductive part? Like same cones. What are the reproductive parts? Cones are the reproductive part. But these cone in netum cones are somewhat different when compared with pinus and other gymnosperms because they are advanced character. Netum reproduce sexually, sexual reproduction mainly. Netum is dioecious, dioecious. That means male and female both are separate. Dioecious, and both the male and female stopella inflorescence are separate. Monoecious means both are same plant but yet different. This inflorescence may be either axillary or terminal. That's not important. Inflorescence composed of strong axis to opposite decosit connect bracts. cup-like structure called cuvule. One, let's discuss one. For example, this A. Let's see A. Male cone. And if you take male cone, it has the axis. This is the axis. This axis differentiate into nodes. This is node, and this is also node, node, and internode, node and internode. After differentiated node and internode, each and every node covered by bracts. This bracts. This is, let's take. This is internode. This is node. Node region group of bracts are there. So this group of cells called as cupule. From this cupules, among the cupules, a ring of arbustu, that means sterile ovules arise with fertile male flower. So these are the male flowers are arised. Male flowers also arise like this. Male flower. If we take the male flower, each male flower have the perianth, stalk, microsporangia. That's it. Microsporangia. And either side uniseriate capitulum present like this. So this is about one male flower. Like pinus, there is no microsporophyll, megasporophyll. Instead of microsporophyll, here simply one layer perianth is present. Perianth is present. So this is called microsporangia. Head region called as microsporangia. Now tell me what are developed inside microsporangia? Hello? What are developed in
Microspores.
microspore? That is common. Only that is structural different in pinus and netum.
Yes.
Likewise, the same female cone also. Same nodes, internodes. Same male and female cone like same axis, same bract cupule, but instead of male flower, female flower. That's it. Male cone already discussed. Once again, we see male stobulus has a long, slender axis. Already told you. This male stobulus is branched or branching maybe several kinds. The axis wears 10 to 12 over bracts, collar or cupule. Don't get confused. This word collar, cupule, same word. From the collar region, about 12 to 25 male flowers are arranged in 3 to 6 rows above the collar. Each collar there are 3 to 6 rings. More male flower, a single ring, and say above the present. Young stobulus is compact due to much reduced axis. Short internodes. Internodes are very short when compared with female cone. Internodes are very short. Continuous. Male flower. The male flower has two unilateral anthera. Already told you with diagrammatically like this. Enclosed small sheathing perianth. It is stalk and antherophore, which elongates maturity. Anther emerges behind the collar through slit into the perianth. Number of male flowers as very. Already we have discussed all things. Microsporangia, microsporogens. What is microsporogens? Already we discussed earlier. What is microsporogenesis? After microsporogenesis, what happened?
Spores
Hello?
are formed.
Right. Spores are formation that is called microsporogenesis. But after production of microspore, this microspore further developed into hello? Spores
Gametes.
further right. Gametophyte first, not direct gametes. Gametophyte, later gametes.
Yes.
So the took hypodermal archisporial cells, multicellular archisporium, repeated division. So these are the archisporium. This is part of microsporangium. Outermost layer archisporial cells divided from primary parietal sporogenous tissue. So these are part of the microsporangium. So anyway, finally, from the sporogenous cells, microspore mother cell developed. This microspore mother cell undergoes to meiosis division and form the microspores. This microspores of further developed into male gametophyte. Next, female gametostobulus. Same here also similar male stobulus. Female stobulus a ring of 4 to 10 female flowers. Same. This is the female cone with ovule or seed. Megasporangium ovule. Let's see the megasporangium or ovule. It is integumented megasporangium is called ovule. It has a stalk. The ovules are orthotropous ovule. The type of ovule orthotropous. Cross nucellus, tenunucellate. I will tell you later. The ovules are remember orthotropous ovule. Cross nucellate type protected by three envelope. The outer envelope is thick, succulent maturity, considered to be perianth. Middle inner envelopes are actually integuments. These are the middle envelope called outer integument. Anatomically similar the outer envelope. The inner envelope, the inner integument elongated or behind the apical left and perianth forms a long micropylar tube and that is called style, so-called style. The inner integument free from nucellar except the collegial. The two sets of vascular bundles are formed which outer, let's see diagrammatically. Here micropyle tube, outer evidence, so middle, inner. So this inner, it shows somewhat extent region like this. This is called this empty gap called micropylar tube. So inside the integument, this is all integument, outer, inner, middle. If you separate the integument, rest of the place called nucellus. The nucellus well developed and it is epidermis divided from nucellar cap. This is clear determination between nucellar cap and parietal tissue. Inside the nucellar tissue, meiosis division takes place as a result formation of megaspore mother cell. These nucellar cells, after division, megaspores are formed. This megaspore further developed into female gametophyte. So gametophyte of natum. Before that, actually, so this is direct the structure. Actually, before the direct structure, let's draw here. Let me draw draft diagram. This, for example, these are the integument, three integument. After integument, let's change color. Only nucellar cells. This is all complete nucellar, no female gametophyte. From the nucellar cells, one is, I don't waste our time. Let me draw rough diagram. Ovule, this new integument, one of the megaspore mother cell. This is MMC megaspore mother cell. Meiosis division 1, 2, 3, 4. One is active, remaining degenerate, and this one is developed as like this. Female gametophyte. Now female gametophyte developed. Inside the female gametophyte, two egg cells are developed like this. Female gamete or egg are developed. So this is the development. This is development. Gametophyte of natum. Already told you microspore male gametophyte, a megaspore female gametophyte. Let's discuss first male gametophyte. Development of male gametophyte before pollination. Microspores is here. This is microspores. Don't get confused here. Microspores here. Microspore. This microspores single first one nucleus microspore. Later it is divided into two nuclei, antheridial cell, prothelial cell. Later again divided into three cells, antheridial cell, prothelial cell, tube cell. Now it is called male gametophyte.
Sir, it
Male
can also
gametophyte.
be said as vegetative cell. Tube
Yes,
cell.
yes, 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 same, same. Actually, in some species, apart from the prothelial cell, vegetative cell also developed. That is mainly formed in angiosperms. That is
Okay.
mainly formed in angiosperms. But the names are different. Arthridial cells, prothelial cells, tube cells. But in some body cells, vegetative cells, mainly if take the angiosperms, only two cells. The two cells are large body cell and vegetative cell. Body cell and vegetative cells. Both are only two developed.
Yes, sir.
Later from the body cells, actual body cell nothing but antheridial cell. From body cells, generative cells, male gametes are developed like that. Here, antheridial cell, one cell is antheridial cell, a prothelial cell, tube cell. At the three cell stage, the pollen grains are released out. Now one nucleus microspore. Now it is developed as a male gametophyte. How many cells in male gametophyte?
Three.
How many? Three cells, right? Now after formation male gametophyte, from the male gametophyte, what are developed?
Male
Male
gamete.
gametes.
That's it. That's it. Now from the tube cell, next from the tube cell, this tube formed, pollen tube formed. From this antheridial cells, two male gametes are developed like this. One and two, two male gametes are developed. So this is development of male gametophyte. Now this is male gamete. Gamete. This is gametophyte. Three-celled gametophyte. Inside the tube, only one cell, that is male gamete. Right? Is it clear? Hello?
Yes. Yes.
Right. Likewise, see the female gametophyte also. Already we discussed just now how the male gametes developed inside the nucellular cells. And let's have again, once again, we check. So this is the development. Take a picture. This is development of male gametophyte. So the different opinions, the development of male gametophyte, Thomson, according to Thomson, different. Here given Thomson opinion that the prothelial cell does not form at all. Here given, as just now someone raised the doubt. The vegetative cell, the same vegetative cell, body cells are also same prothelial cell. So here that is the opinion of the scientist. For example, Thomson. Thomson, according to Thomson, prothelial cell does not form in male gametophyte. That is opinion of Thomson. The microspore nucellus divide and nucellular and generative cell here. Generative cell later divided into stalk cell, body cell. Here you can see tube nuclear cell, body center. The pollen tube body divides equal male gametes. According to Nagy and Madhulata, the microspore nucellus netum Niemann Hola divides small lenticellular cells and a large cell. The lenticellular does not take part in further development, ultimately disappear. But they did not give, they did not mention any prothelial cell, any body cell, any vegetative cell. Simple, they mention lenticellular cells and a large cell. Simple, two. Now, female gametophyte, megasporogenesis, same male, female gametophyte also we discussed earlier. Uh, how the female gametophyte development inside the archis nucellular cells. First of all, in nucellular cells, megaspore mother cell, megasporogenesis happen. From megasporogenesis, female gametophyte develop like this. Right. After development to female gametophyte, pollination. Pollination, already transfer of male gametes towards the female gamete. That is wind pollination. Now the pollen grain shed at three-celled stage, three nuclear stage. Already told you. By pollination drop when reach the female gametophyte or ovule, it is settled as pollination drop, which rich in sugar and executed the top exclusion. As the fluid dries, the pollen grain sunk into the micropylar canal and fused with female gamete. Already we know the development of male gametophyte. Let me discuss. What is fertilization? Fusion of male gamete with female gamete, that is fertilization. Due to fertilization, what happened? In the embryo formation, this embryo developed as a seed. Seed germinated as a sporophyte. Like this. This is the germination, seed germination. Coated and leaf formed. Life cycle important. Take this picture. Life. What we discussed so far, and it is give a graphical pattern that is called a life cycle. If you see this life cycle, we start the class with sporophytic plant body. Let's take. So this is sporophytic plant body starting sporophytic plant body. Start with this sporophytic plant body root and shoot leaf are there. You can see here also male cone, female cone, male cone, microsporophyll. Actually, microsporophyll, but better word is direct male flower. Male flower. From the male flower, microspores are arranged. Likewise, here also female cone, megaspore, one of the megaspore, functional megaspore. From this functional megaspore, female gametophyte developed inside the nucellar cell. Microspore further developed into male gametophyte. From the male gametophyte, male gametes are formed. Later, later, female gametophyte. Inside female gamete, egg or female gamete, male gamete fuses fertilization. Now, until it is gametophytic stage, only gametes are formed. So that why it is gametophytic. After fertilization, zygote, sporophytic. Zygote, this zygote further developed into embryo. Embryo further developed into seed. And again, seed developed into sporophyte. Simple, that's it. We start with the sporophyte and again end with the sporophytic. Take this picture also. Is a clear natum life cycle and economic importance you can read yourself. Any doubt in pinus and natum? Hello?
No, sir.
No, sir.
Clear?
Yes.
All class completed, I think. Any doubt in second semester? Are you attending practical training classes?
Yes,
Yes,
sir.
sir. Done.
Right. Almost in practical training classes, most of the points are completed about bryophyta, about pteridophyta, about gymnosperms. So if you have any doubt, ask me, and again we will discuss in training class. Okay?
Okay, sir.
Right. Can I quit the class? Can I
Yeah,
quit
sir.
or I did?
Thank you so much.
Okay, right. Did you take this picture, life cycle? Let make it clear.
Yes, sir.
Life cycle very important. Life cycle flow chart important. Right. This is life cycle. It is not a. It is no need to draw diagram. You can write the names also. That means sporophyte, microsporophyll, male flower, female flower, male gamete, female gamete. That's it. Egg cell. No diagrammatic. Only you can write with the names. Names also life cycle clear. And remember and economic importance also you can see. Okay.
Sir, paleobotany we have to read by ourselves.
Which one?
Last unit, paleobotany.
Paleobotany. Right, right. Actually, the lack of enough time, we did not unable to explain that. Paleobotany also important.
Okay, sir.
Paleobotany block. Here the each and every block important. Four block there in second semester. Uh,
Yes, sir.
the paper development, paper blueprint from each and every block should be covered. That's it. You should read paleobotany also. In paleobotany, mainly type of fossil, fossilization, main fossilization, types of fossil, and Benitez characters, also, Williamsonian characters. These are
Yes.
important. Please, tomorrow, that Monday, we will discuss in training classes about that. If you have any doubt, ask me. You can read yourself. First of all,
Yes.
this is very easy. If you unable to understand about this paleobotany, inform me. I will explain. Okay.
Okay, sir. Thank you.
Okay. Right. Thank you. Thank you all.